officially I'm going to go ahead and share my screen. Is there anyone who's having trouble seeing my screen? Okay, and just a reminder, if you just got back to your computer, please type back in the chat so I can make sure everyone is back. We might just be waiting for one more. If you guys can all check your SMCE instance, look at the bottom corner down here that I have highlighted and make sure that it says Python conda environment :GL4U, and it should also say that it's idle. Is there anyone who's not seeing that? Okay, great. So before we went to lunch, we had just taken a look at the standard, we submitted our star alignment array Slurm script to run this star command for all 12 of our samples. And I know I went over all of these options in the lecture, but is there anyone who wants me to go over what any of these options mean again? Okay. And so we went ahead and submitted our array Slurm script to run that command on all 12 samples. And then just before we left for our break, we took a look at the standard output from the star command. And I just kind of quickly went through what the star command should output. And so I do just have a couple of questions. Lisa, Armin, if you're on, if you could queue these up and put the link in the chat again for everyone. So we'll answer the first one first, which is, did the star alignment complete successfully, and how do you know based on the standard star output? You can go ahead and type your answer in the box and the link that Lisa put in the chat. So answer the question, did the star alignment complete successfully? If so, how do you know? Or if not, how do you know? And hit submit in the link that Lisa put in the chat once you've written out your answer to the question.
If any of you don't see that question right now, you can hover over the top of the page and it will send you to the slide that's currently being presented, which is that question.
And how are we doing on answers, Lisa?
$8. I think we're missing one.
Just give another few seconds.
Oh, we're good.
Awesome. Okay, copy paste finished successfully. Yes, it tells us. Yes, it says. Yep, it says so. Yeah, yeah, it says so. Standard up. Yeah, exactly. So pretty nice, convenient that this tool tells you that it has finished and that it finished successfully. So again, if you were to be processing your own data using star, if you saw anything other than this in your output, anything other than the finished successfully, or if there's an error message in there, you would know that you have to go back and troubleshoot. And then next question here is, how long did it take for the star alignment to complete? So Lisa, if you want to put that question up. So how long did this take to complete for this one sample? So we're just looking at the standard output from one of the samples here. How are we doing on the answers, Lisa?
We have 8.
Okay.
We're good.
Okay, great. 1.9 hours, just under 2 hours, hour and 57 minutes, 1.96, almost 2 hours, 0.0002% of a year. Yeah, so we know this a couple of ways. So I add timestamps to the start and end of all of my scripts to keep track of this. So you could have just used that, which I think a lot of you did. The other thing that's nice about STAR is it tells you, it gives you timestamps in kind of the standard output. So it tells you when it starts and then it gives you a timestamp for when it ends. So that's another way that you could tell just from the STAR output itself without the extra stuff that I had in there. And just to kind of show you guys, this was 2 hours. And if we take a look at the script that you guys all submitted, again, what command do we use to view files? Type it in the chat. Yeah, Mark, we use the cat command. So if we just want to take a quick look at this file, we can see how many resources that we gave it per job. And I did copy and paste that command. So if you just want to add a code block, copy and paste the command that I put in the chat. We should see this. And again, with using the Slurm script, we allocate how many resources we want to give each job. So you'll notice here the memory is in megabytes. So I gave it 60,000 megabytes or 60 gigabytes and also 18 CPUs. So with 60 gigabytes and 18 CPUs for this particular sample, it took about 2 hours. And so this will be faster or slower depending on the number of resources that you have available. So this is, again, the most computationally heavy step of the pipeline. Okay, now that we know how it was run and that it ran successfully, we're going to take a look at some of these other outputs, starting with the actual BAM file. This, we're going to be looking at the align to transcriptome out BAM file. And again, this is going to be alignment information for every single read. So we know that these reads range from about 65 million to about 85 million reads, and that each read could map to multiple locations on the genome. And so we're going to go ahead and take a look. We're going to go ahead and take a look at the first few lines of this BAM file. So I want everyone to go ahead and run that. And I just say again, this is the BAM file indicating the alignment to a transcriptome. Remember, we outputted 2 alignment files or 2 BAM files. One, it was aligned to the genome, so we would have gene IDs. This one is aligned to the transcriptome. So we have transcript IDs, all ensemble IDs, because we used an ensemble reference FASTA and GTF file when we did our alignment. And so I went over which each column of this file is in the lecture. So here in the lecture is where I went over. This is what we're looking at, is the aligned to transcriptome BAM file. And I do also want to point out how we normally use cat to view files. You'll notice that here we're using a file or a tool called SAM tools. So as a reminder, BAM files are compressed binary versions of sequence alignment map or SAM files. So they need to be uncompressed, but they need to be uncompressed in the special way that was used to compress them. And so there's a tool called SAM tools. SAM tools is a great tool. It has a lot of different functionalities. And so one of the functions that we're using of SAM tools is called view. And this one will allow you to view BAM files. And then again, because this contains 65 to 85 million reads, depending on which sample we're looking at. We obviously don't want to see all of them, and it could even be more lines than that because each read could map to multiple locations on the genome. So rather than opening all of it up with SAM Tools View, we're going to just open up enough for the head function, which allows you to look at the first 10 lines of the file. So when we take a look at the first 10 lines of this flight rep 1 alignment file, aligned to transcriptome file, we see those different columns. So at least 11 of these fields are going to be present in your BAM file. And I listed all of these fields again here in the Jupyter Notebook. So again, you have this information both in the lecture as well as right in the Jupyter Notebook. And so I'm going to ask you to take a look at that BAM file and answer the following questions. And so you can remind yourself what each of these columns mean by reading through these definitions. If any of the definitions are unclear to you, let me know. This is probably to writing standard output failed. Yeah, Elba, that is a problem. Is anyone else not seeing this? When they do their SAM Tools
It was
View?
at the end of the file, so it printed the beginning and then it gave that.
Yeah, that's fine. So this is anytime, right? So SAM Tools View means that it's going to try to print out every single line of the file. So when I piped it into head, as soon as it went into the head command, after it showed those 10 lines, which is what head allows, but head stops showing things after the first 10 lines. But SAM Tools View is still trying to make them be print out. So when it runs into this head command that stops it after the 10 lines, the SAM Tools View will say that it had to stop writing after those lines because you broke that tool by piping it into head.
So that's to be expected.
Yeah, to be expected. Yeah, we saw a similar thing up here. We were opening up a file with cat, and you saw a similar thing. I think we were opening up our FastQ files before the FastQC that we did. We opened up a couple of these FastQ files with zcat, the special sister command of cat that allows you to look at gzipped compressed files, and we piped that into head. And you'll notice that at the end of that command, gzip tells us that it was broken too, because again, head only allows it to show 10 lines. And so once it reaches those 10 lines, head won't let it print out anymore. So it like stops, it breaks that command from running. And so you get that error from that command. So same thing happens with SAM Tools View when you pipe it into head. As soon as it's done printing out those 10 lines, head doesn't allow it to print anymore. So it essentially breaks the command that you ran initially. So now taking a look at this BAM file, again, each line here. So this is all one line. Each line starts with the read header. So this line goes from here all the way across here. So that full thing, and you can, it goes down to the next line because it's only going to print as big as the view is. But this is one read alignment information. This is the next one. All the way through, this is the next one. So they each start with the name, the read header is the first line. So this is going to be showing 10 because again, we piped it into head. So it's going to be showing the results from 10 different reads. So Lisa, if you got those questions queued up, the first question I want you guys to answer is how many transcripts are represented in the BAM file and how do you know? So how many transcripts do you see represented in the first 10 lines of this BAM file? And how do you know? And let me know what is easier for me to have on my screen. If you want me to have up the definition of the lines, if you want me to show the actual output of the BAM file or the question, or if it doesn't matter. Yeah, how many transcripts do you see in that first 10 lines of the BAM file? And how do you know? How are we doing on answers, Lisa?
Oh, just waiting for one more.
So we just need one more answer.
Got it.
Awesome. So we have 5. There are 5 unique values in the third column of the first 10 lines. 2 transcripts looking at the read names. You said 2 transcripts, but then you're looking at the read names. Wouldn't that be 2 read names? 5, there are 5 unique ensemble transcript IDs, and said 10. 10, I control F and highlight. The underlying format is that each entry is a single row. We got 5, 10, unless I can't count. 5, number of ensemble. 10 because I counted and because we use the head command, which reports the first 10 lines. 10, count the Q name. 5 trans. Okay, so we have a lot of mix of 5 and 10 with 1, 2 here. So 2, you're actually getting to another question that are asked. But yeah, so transcripts, you want to look. So you guys are right. We do print the first 10 lines of the file, right? So we are seeing the first 10 lines of the file. But in terms of how many transcripts we're seeing, which column tells us transcript names or transcript IDs? You can type it in the chat. Which column tells us transcript names? Again, because this isn't aligned to transcriptome file. Yeah, our name. So the third column here is the reference name of the reference sequence the read is aligned to. This will be ensemble transcript ID in the transcript aligned BAM file, which is what we're seeing above. If this was the aligned to genome file, it would be the chromosome number. So again, because we're looking at the aligned to transcript BAM file, that R name in that third column is going to represent the ensemble transcript ID. And so that's what you want to look for to know how many transcripts there are. And so just like some of you guys mentioned in your answer, we're going to go through this third column and look. I think I can highlight more than one thing. No, we're going to look at every single ID here. So that and that is the same. So far we just have 1. This is different than that. So now we have 2. This is the same as the one above it. So still just 2. This is different than the 2 above it. So now we're up to 3. This is the same as the one above it. So still 3. This ID is different than any of the ones above it, so now we're at 4. This one is the same as the one above it, so we're still at 4. This one is different than any of the ones above it, so now we have 5 so far. And then looking at this last line here, that's the same as the one above it. So 5 total transcripts we see in the first 10 lines of this file. Is anyone confused still about how we know how many transcripts are being identified? Okay, great. So we're going to move on to the next question, which is how many read pairs are shown and how do you know? So I talked about this a little bit in the lecture. So you guys go ahead and answer that next question. How many read pairs are shown and how do you know?
I have a bit of an unrelated side question.
Sure.
So one of the problems that I kind of face that makes it kind of that slows me down when answering these questions is that whenever I go from split screen on the Jupyter Hub to full screen, it completely shifts where I am on the lines. So like I would be reading something and then I would, because I'm working on a laptop right now, I would need to go full screen in order to answer the questions. And then it will just shift me like 30 lines up or 30% up to the top of the page. And so I have to re-scroll all the way down just to find where the answer was. So I don't know if there's a fix for that or whether I'm just setting it up wrong.
I don't know if there's a fix for that. I know that this is hard to do with just one screen. That's kind of some feedback we've gotten in the past because it's back and forth with the questions. The other thing we could do is we could go back to polling within WebEx. The reason I went,
So,
go ahead.
one other idea, Amanda, is that there is a way to see an interactive table of contents on the left. If you go all the way to the left of your screen, Amanda, and click on yeah. So if you have that visible, everybody, then if you lose your place, you might be able to find it more easily.
So I moved away from the screen at that instant because I have to go back and forth between your slides, Amanda, because I have a very small computer for travel and the other one, so that slows me down. So what did you just click on? If you go back, what did you?
Sure. So on this left-hand panel, we're usually here. So all the way on the
Yes.
left-hand side of your Jupyter Hub, if you go all the way top and click the folder, that's showing like
That's
our
that
home
view.
directory where we are. But if you scroll down in the one before the last, the one above the puzzle piece,
Perfect.
bullet points will give you the
I
table
love
of
it.
contents.
Thank you. Thank you.
Thanks, Lauren,
Yeah,
for that tip.
thank you. That makes a huge difference. I'm so happy that I'm not the only one that's having this small screen issue.
No, mine is tiny. I
Mine is
travel light.
mine is 13 inches. It's pretty small too.
That's about mine. Mine might be 12. It's a small light computer.
Yeah, thanks for that tip, Lauren. Yeah, sorry about that, guys. I know this virtual life sucks.
Is Mentimeter forcing your screen to go to full screen?
I mean, it's not that. It's the fact that I'm trying to juggle the slides,
Oh,
this
okay.
meeting, the voting poll, and the Jupyter
Yeah.
Hub, four windows inside, 13 inches, and so
Right.
I'm like alt tabbing like a gamer. Like it's still difficult.
That's what I'm doing. Exactly. I have one full screen of 3 different locations and I go back and forth between them.
Yeah. I have a Mac, and my favorite feature on the Mac is the four-finger swipe up to show you everything on your screen, and that's usually how I navigate when I have multiple things.
You're so cool.
Okay, I'm gonna go ahead and show the answer. I don't know if all of you guys have finished yet, but we'll go over it together for those of you who haven't gotten it yet. So 2 for each with a question mark, 2 with no explanation, 2, there are 2 unique entries in the first column, 5 read pairs, their IDs are the same, and we're taking the forward and reverse primers of these transcripts, interesting, 5 based on the transcript name. So remember, we're looking at reads now. So I know that I got an answer last time that said the number of transcripts, but said that they got the number of transcripts by looking at the read name, and now I'm seeing an answer 5 for the number of reads based on the transcript name. So remember, keep in mind the question that we're asking. I can't tell, fair enough. 5 share the first column. 2 read pairs, 2 pairs have 3 for SAM code. 5, the read pairs have the same ID but different flag. Okay, let's go over this. Yeah, this one's hard. A lot of people don't often get this who are unfamiliar with these files. So keep in mind that first column is going to be the query template name. So that's going to be the information, as we went over in the lecture, about the read, right? So this is actually part of the read header. It's the first part of the read header here. And so if you remember, this is essentially an address for a cluster on the flow cell, right? And so if you have a read pair, your forward and your reverse read, because it's dealing with the same insert or same template that it's sequencing, so it's going to have the same address. It's going to have the same cluster address. So the first thing that we're looking for is how many cluster addresses do we have here? And so it looks like this one and this one are the same, right? And then, so that's either the same template, either forward or reverse, right? We have another one here that is the same as the 2 above it, right? Same exact read information, so same cluster location. So again, same template, still either forward or reverse of that template. And then we have another one here, same exact address. So it's the same insert or template whatever terminology you want to use. I hear both. That's why I keep using those interchangeably. But again, either forward or reverse. Don't know that yet from here. Have this one here. Okay, this one's different now. So far we have one template name, and now this is our second template name. And then if we look at the next one, that's the same as the one above it, so still just 2 templates. And then this one here is the same as the one above it, so still just 2 templates. Another one here, that's the same as one above it, so still only 2. Another one here, again, same as the one above it, still only 2. And another one here is the same as the one above it, so still only 2, right? So we know 2 templates. Still don't know from that if they're forward or reverse. So for that, we can go into either the position, I think would be the one that I would go with. No, not position. The template, probably template length. Yeah. So the template length is going to be the length of your insert, right? So the leftmost position of read 1 to the rightmost position of read 2. And depending on the orientation of that relative to the reference, it's either going to be positive or negative. And so keeping in mind that when you do paired end sequencing, you're going to be sequencing in one direction and then in the opposite direction. So if you have a mate pair, you're going to have the same query name. So the same read name here, identifying that they're from the same cluster location. And what you should also see is that they'll have the same, since this is broken apart, the same templates on this column. I know it's split up here on my screen. I don't know, yours might be different. I change the size of mine, make that easier to see. Yeah, that's better. So here we have the same template name for this one and this one, or query name, excuse me, the same query name for this one and this one, or same cluster location, cluster address. And you see that this template length is positive relative to the reference. And this template length is the same as the one above it, but it's negative relative to the reference. And so since your forward and your reverse reads are acting on the same template, just in opposite directions, that's how you know if it's a mate pair. So we have the same cluster location in both of these, and it's the same template length, but one's in one direction and one's in the opposite direction. So these must be a mate pair, forward and a reverse read. And then if we go down to the next one here, this is the same exact cluster as the one above it. And then if we look at the template, we see it 167 in the positive. So this read must be the same as this read in the same direction. And then if we look at the one below that, again, same cluster location, but in the opposite direction. So these two, the first 2 lines and lines 3 and 4 are going to represent the same cluster location, forward and reverse, 2 different transcripts, right? Because you see 2 different transcript IDs here. So this mate pair here is mapping to 2 different locations on the reference, right? This first location here we see is mapped to this transcript ID. The second location here, that forward and that respective reverse read, is mapping to a different transcript than what that same pair is indicated above it. Starting to answer some of our next questions. But then this one here, we see it's a unique read header from the first 4 lines. And so this is a different location on our flow cell. So it represents a different cluster or a different insert or template, whichever word you want to use. And we see that we see the template length is 203, and it's in the positive direction with respect to the reference. And the one just below it has the same location on the cluster, so the same address there. So we know that this is sequencing a sequence from that same template as the one above it, and which makes sense because it has the same template length, but this is in the opposite direction as the one above it. So again, these 2 here represent a mate pair, and we see the transcript that it is aligning to. And then just below that, so on line 7 here, we have again the same exact address, right? The same cluster location. And this one is again template length of 203 in the positive orientation with respect to the reference. So this read here is going to be the same as this read here. And then just below that, again, has the same address, the same cluster location, but this time it's in the same template length, but this time in the opposite direction. So again, mate pair, and this read here is the same one as this read here. And so what we saw up here is that this read right here is mapping in the forward and the reverse direction to this transcript, right? See the same transcript name. And then that same mate pair, so same forward and reverse read for that same template, is also mapping to this transcript here, right? Same transcript name with that same mate pair, so that's different than this transcript, so mapping to two different transcripts. And then if we look at line 9, we see that this read header has the same address, is several ones above it, right? Is this one and this one and this one and that one. And then if we look at the template length is 203 and it's in the positive direction. So this again is the third time we're seeing this read. And then if you look at the one below that, has the same cluster location or address as the one above it. So again, working on the same template or insert, however you want to think about it. And same template length, except this time in the negative direction. So this is now the third time that we're seeing this read, which is the opposite orientation of the one above it, acting on that same template. And what we see with these last two lines, 9 and 10, is that they're mapping to a transcript that's different than this one or this one. So that would be the third transcript that we're seeing this mate pair map to. Any questions about that? Or how we identified that there are only 2 mate pairs that we're seeing in the first 10 lines of this file?
So tell me again what the cluster is, because you're saying they're all in the same cluster and yet they're mapping to different, reference genomes. So this then is the way we're trying to get these two reconciled. And then the fact they're the same length doesn't mean they're complementary, because I thought that we were not having much overlap, correct? But we trimmed them so they should be the same length. That helps find them. Is that right, Amanda? Am I completely messed up here?
Good questions. So first about the clusters. So keep in mind when we do the sequencing, we have prepared these libraries, right? So this is what I've been referring to as our template, the sequence of interest, right? And so that's going to latch onto the flow cell. We're going to have that bridge amplification. It's going to form these clusters, which are all exact copies of that same sequence. And we're going to sequence it in one direction, and then we're going to wash everything off and sequence the same exact sequence, just in the opposite direction, right? So the location of this cluster on the flow cell, like the address of it, that's what I'm referring to when we look at this. So this information is like flow cell number and the location.
Yes, exactly.
Right? So we know that these are from the same cluster.
Yeah.
The reason that we're seeing the template length being the same, because you're right, the size of the read, right, the length of the read is not necessarily the same for forward and reverse. The example I made has them the same, but that doesn't have to be true. But
Okay.
so if we look at this example here, right, let's say we have, this is our sequence that we're sequencing, right? This is our insert or our template that we're sequencing. So this is the reference genome, right?
Okay.
And so when we go to align these reads, our forward read here I've labeled as R1 in red, our reverse read here I have labeled in blue is R2. And so what the alignment tool is doing is it is identifying where it maps to on the reference genome. And so in this example here, we have R1 starting in the leftmost position at 10 in this black reference that we have here. And it goes to location 160. And then in this example, we have our reverse read mapping to, in the opposite direction, 280 on the 3' end and 130 on the 5' end. And so it always lists the orientation of the insert in the 5' to 3' direction. And so it's going to list that R2 in the reverse, so it'll be a negative value for the total length. So this is your template. And then in your R1, it's reading this way, and it knows to go to R2 because it has that same cluster location, so that it'll go to the end of R2, the right side of R2, and that's going to give you your template length for this read and for this read. Remember, they're thought of together because they're a pair. And so the template length corresponding to R1 is going to be 270 in total. The template length corresponding to R2 is going to be 270 in total because those reads come together as a pair. And so that's why it's the same template length that we have. The only difference is whether or not this 270 is going to be positive. So reading from, if the forward read is in the same orientation, 5' to 3' in this case, of the template, then that's 270 is going to be a positive value. If the read is focusing on a template that's in the opposite orientation relative to the reference, that template length is going to be a negative value. Does that make sense? Still not clear.
It's a lot of assumptions the program is making for you.
Not assumptions. It knows that these two reads belong together because it has the same exact cluster information. And so it knows that this read and this read go together because their address is exactly the same, right? Their address is the same. And then it's looking at the orientation. So it does a template length first we told it that it's paired end here because we gave it a forward and a reverse read. And so the tool knows that this is paired end. So with any read, it's always looking for its mate pair to determine what the template length is. And then based on the orientation, it's just looking at how this sequence maps back to the reference, and if it's going to be in the same orientation as the reference, or if it maps back to the reverse complement orientation of the reference. And so because you have a forward and reverse read, it's going to act on the same template, which you know is the same because, again, you're given the same address in the forward and the reverse read. It knows the size of that template because it starts at the leftmost position of the forward read and it ends at the rightmost position of where the reverse read maps. And it's always going to map it with respect of the reference genome in the 5' to 3' direction. And so when it goes to align it, it will try to align it in the 5' to 3' orientation, also in the, if it would be the reverse complement of that orientation. And so when you have a paired end read, one of them is going to be the reverse complement of the other with respect to the template that it aligns to.
So Amanda, I'm a little confused as to how there's only 2 read pairs.
Okay. So are you understanding the information that's provided in the read header here?
Yes.
Okay, so this is essentially an address that's going to correspond to the cluster location that we're sequencing, right? So to this location here. So that means that that address is going to be the same for both the forward and the reverse read acting on that template in that cluster. You follow that?
Yes, and that's a read pair, right?
The forward and the reverse read is considered a read pair, and it would be acting on a template because it came from when you chopped up your RNA here. So you have your RNA and you cut it up, right? So each piece that you're sequencing, you're going to sequence it. This is going to be your template, and you're going to sequence that template in both directions. So one forward and one reverse. And so for this template, it's going to have a mate pair, one that sequenced the template in forward direction and one that sequenced the template in the reverse direction, sequencing that same template, right? It's in the same location on the flow cell. And so the address of where it resides on the flow cell, the template that's being sequenced, is going to be provided in that first line of the read header. So this is a location information for the template that's being sequenced. And STAR knows, because you gave it a forward and reverse pair, that it's expecting there to be 2 for every read header that it sees, right? So for every one of these unique addresses, it should see 2 addresses, one for the forward read and one for the reverse read. So it analyzes these mate pairs together. And what this is looking at is that template that's being sequenced. So again, if I go back here, this here is the template that's being sequenced in the forward and in the reverse direction. And so when it goes to map it back to the reference genome. So here's our reference genome. It's going to look for where this sequence maps best to this reference genome, and it's going to check both directions. So it's going to see how this maps back to this reference genome, if it's in this direction, or if it were the reverse complement of that direction. So how it orients itself relative to the reference. And so your template in this case here is green. So again, that green bar is going to have the same address in the read header for both sequencing it in this direction and this direction. So the first thing it does is it identifies these read pairs. And then once it identifies these read pairs, it sees where they map to on the reference genome. And then once it knows where this and this are together, they both map to this location in this example on the reference genome. So then it's going to see how long this template is it was being sequenced. And again, the template coming, this being the template, right? So we don't know how long that is. We just know how long our forward read is and how long our reverse read is, right? But that insert that it's sequencing in the forward and reverse direction, we don't know how long that is yet until we map our forward and our reverse read. And so once we identify those read pairs, again, because they have the same address in their read header, now we're going to see where they map to. So we see in this example that read 1 maps to starting at position 10 to position 160 on this reference genome depicted here in black. And we know that its mate pair, again, because it has the same address, its mate pair here maps to position 130 to 280 on this reference, but the mapping, it actually maps in the reverse complement orientation to that position. So what it does is it aligns it in the reverse complement, actually. That's why these are pointing in different ways. And then it says, okay, the leftmost position of R1 here is going to be the start of my template length, and the end of my template length is going to be the end of R2 when it's mapping in the reverse complement orientation of its respective mate pair. And so the start leftmost position of read 1 and the rightmost position of where its mate pair maps is going to be the total length of the original piece of RNA that was sequenced. So that, if I'm saying this is the same example, so that 270 length would be this total length here. Even though, as you see in the example, so we don't know that until we map this. So right now, all that we knew was the length of read 1, it being 150 base pairs. And this one, it just so happens that read 2 in the example I used here is also 150 base pairs. But again, that read 1 and read 2 length don't necessarily have to be the same. But we just know that they go together because they have the same address. So we'll see which one of those reads maps to the reference genome in the same orientation as the reference genome, and that'll be considered the forward read or the first read here, where it's going to start the template length at the leftmost position. And then if this read 1 in this case, is mapping in the same orientation as the reference, then it knows that its mate, again with the same address, should be mapping in the reverse complement orientation to another place further downstream. And so it'll look for where that maps, and then at the rightmost position of where that's mapping in the reverse orientation with respect to the genome is where the end of the template length is going to be. Is that
So basically this hinges on the fact that you feed the data with the cluster location and say, these two go together, make it work by using this kind of analysis against the reference genome.
right? Yeah, you tell them
It hinges on the fact that you're feeding it the cluster location and saying these two are a pair.
right.
And then it finds, I guess, the best solution that
It
matches both of them to a transcript.
It finds where each one of them map to the transcript, and then it tells you the link. So then another thing to your question of where it how it can find or how it can map to more than one location. So
Yeah.
what I'm showing here is just it mapping to one location, right? This is just one location on the reference. However, this can map to another location on the reference, right? So it can, maybe there's a sequence downstream over here on another transcript that has a very similar sequence in the area covered by the forward read and the area covered by the reverse read. And so if it has a really similar sequence somewhere else in the genome, then your tool, as long as it matches within the amount of errors you allow it. So what GeneLab does is we allow a 4 base pair mismatch for every 100 base pair read length. So as long as it maps within that criteria to anywhere on the transcript, anywhere on the transcriptome, in this case we're looking at a line transcriptome, then it'll show up in the BAM file. So now if we have something like this, what we'll get back in the BAM file is we'll get our mate pair here with this template length mapping to these genomic coordinates here and here. These should be different numbers. Let's just add zeros at the end of this, of all of these. That way it looks like a different location. So now in this example, these mate pairs, right? Again, the same exact mate pairs. We see that the forward read aligns to both this location on the reference genome, but it also aligns to this location on the reference genome. Again, because there's similarity in those two locations on the reference genome in the sequences. And we also see that this reverse read maps to 2 different locations on the genome. And so again, the forward and reverse go together. So it'll look at the start of this one, and then it'll go to find its mate pair and the closest one that maps to it, right? So these ones are going to be, this forward read is going to be closer to this reverse read mapping location than this one. So it'll look for the first one of its mate pairs, again, that has the same address of where it aligns to next. So that's why this one will go with this R2 mapping to the same transcript, one location. But then we also see that the forward and reverse reads can map to another location on another transcript somewhere else in the genome. And so we'll also see that in the BAM file that we have the same insert. And again, the template length isn't going to change, right? Because we still have that same insert that we're mapping. We're just trying to figure out where it came from. So it didn't come from both of these locations, right? But we don't know which location it came from yet. We may not ever know which location for sure that it came from because there's sequence similarity in the genome and it maps to both of these locations. And so when we go and we look at our BAM file, now we see if we take this first mate pair here, so this mate pair, we know it's a mate pair again because it has the same address, right? The same cluster location. It has the same template length. Again, it has to have the same template length because we're sequencing the same template, just one in the forward and one in the reverse. So it should have the same template length as we see here. We just see opposite orientation relative to the reference. Because again, in this example, our forward read is aligning in the same direction, and our reverse read, it aligns in the opposite orientation. So that's represented in the BAM file with a negative symbol in front of its template length. So these are the same exact template, just one isn't looking at the forward read and one is looking at the reverse read. And then the transcript that it's aligning to, or the location where it's mapping to on the reference genome, is this transcript here that ends in 190277. But then we also see that that same mate pair, again, we know it's the same mate pair because it has the same address, the same cluster location as the one as the mate pair above it. We also see it has the same template length, right? Because again, it's the same sequence. We're just we're like laying it down on the reference genome in various locations to kind of see where it aligns to. And because of the redundancy in the genome, it can align to multiple locations. So what we're seeing again here is the same mate pair, which is the sequence of this same template that's in this case 167 base pairs long. And the forward read is going to be mapping in the same orientation relative to the reference in this case. The reverse read is going to be mapping to in the opposite orientation relative to the reference. So that's why you have a positive sign here for the template link and a negative sign. And then, but in this case, it's mapping to this transcript that ends in 82409. So again, same mate pair. So we have one mate pair here that's mapping to this transcript, and that same mate pair is also mapping to this transcript. So this is a multi-mapped read because it's mapped to multiple locations on the genome. In this case, based on what we're seeing so far, this mate pair, which again is representing the forward and the reverse sequence of the same template, is mapping to 2 different locations on the genome, 2 different transcripts. And then what we see here in the last 6 lines of this BAM file is we see another mate pair. Again, we know because we see the same address for every single one of these last 6 lines, and also the same template length, just one associated with one read in the positive direction relative to the reference. It's mate pair associated with the reverse complement orientation relative to the reference. So same template, same address, just different sign. That's how we know that they're mate pairs. And so we see them going order, and they go in order here because we tell it to sort, so it should also sort the mate pairs too. So we see the forward mate and the reverse mate acting on a template that's length 203. So in that case, this size would be 203 instead of 207. And we see here that it maps to transcript 7038. But then the same mate pair, again, same address, same template length, and then these two that come right after each other have just different orientation of template lengths. But we see that it's a different transcript here. So for every transcript that it maps to, each read will be a new line in your BAM file. So far what we see here, and I'm giving away the answer to the next question, but so far we see here for whereas this first mate pair, right, we saw map to 2 different transcripts. It maps, this mate pair maps to this transcript, and this mate pair also maps to this transcript. And now the second mate pair that we're seeing, and again, we know we're seeing 2 mate pairs because we only see 2 unique cluster IDs, and they each in pair are either a positive or negative template length. And again, same template length would correspond to the same insert template that's being sequenced. And in this case, for this one, we have the mate pair is going to maps to this location, so to this transcript. And then this same mate pair, again, same address, same template length, and the mates are there, positive and negative. But this time we see it mapping to a different location on the genome, a different transcript. And then we actually even see the same mate pair again, we know that because the same address, same template length, just different orientation with respect to the reference, but it's mapping to even a third location. And so are there any other questions about why we're seeing 2 mate pairs only here, how we know? And it's mapping to multiple, both of these mate pairs are mapping to more than one location on the reference genome. They're mapping to more than one transcript.
This is a great example. It's clarified so much. I love it. But when you look at the map quality, which I guess you can look at, some have 0s and some have 3s. Is that going to help with the tiebreaker, or is this basically I mean, how do you reconcile that? Do you care? Do you say it maps to everything?
Good
Because
question.
presumably there was one transcript it came from when you sequenced.
Right.
So
Yeah. So there was only one transcript that came from when we sequenced. We're just this is a puzzle now. We're just trying to figure
yeah.
out where these puzzle pieces came from. And yeah, so the map quality does have something to do with it. So if it uses a 255 if it maps to
Yeah, 0255.
yeah. So 3, I'm pretty sure, not 100% sure, but I think, so these numbers relate to how many mismatches in the template length, right? Because again, if you have something that in this example is 270, I'm gonna make it easier for myself and pretend like this is 200. So if this template was 200 and we allow, well, I guess no, these read lengths are fine. So these read lengths are 150 each, and we allow for up to 4% mismatch. So 150 would mean that it could have up to 6 mismatches, right? So there could be 6 base pairs in this read and up to 6 base pairs in this read that don't match exactly. And the location will still accept it as a mapping. And so how many mismatches it has from the read to the reference will be encompassed within this mapping quality score. And so you can use that as an assessment, but what you'll notice here for all of these, right? So this, and I don't remember what exactly the 3 is. I'm gonna have to go back and look at the documentation, but it's equal to the negative 10 base of the probability that the mapping position is wrong based on the number of mismatches. And so you'll see that these all have the same, this transcript here. So this mate pair that maps to this transcript has the same mapping quality as it did when it mapped to this transcript. So that doesn't give you too much more useful information. And like the same thing is true for this too. So this other mate pair that maps to 3 different locations, we see that for each one, so for this transcript, map score is 0. For this transcript, map score is 0 for this transcript, it's also 0. And so there's no difference in the mapping quality, which means there's no difference in the number of base pairs that were mismatched in the forward and the reverse read relative to the reference. So sometimes it can help, because sometimes it's the case where one location, it has more of an exact match than another location, but not always. Sometimes it'll be the same number of matches or not in the other location. So little tricky. So we'll get into how we do with multi-mapped reads when we talk about quantitation. So we'll get into that a little more. Any other questions though about identifying mate pairs in a BAM file? Identifying transcripts that mate pairs are mapping to in a BAM file? Is it clear now? Because if not, I could also bring on one of the other instructors. Everyone explains things differently.
No, I got it now. When you said that it could map to multiple locations, that was like the light bulb that went off in my head.
Okay,
Yeah.
great. Yeah, no, guys, these files trip me up for a long time. And like when you're going back and trying, if you run into errors, or like I was testing tools out and things weren't looking right, and I had to go back and really kind of troubleshoot by trying to figure out what's going on with the mate pairs in the BAM file. It takes a while to wrap your head around the BAM file and how things are laid out there, but it's super useful when, especially when you're troubleshooting things or need to know what's going on or what you're seeing. Okay,
Well,
so
your example helps connect it to the sequencing, so I love it. It's really a beautiful presentation going back and forth. It puts the experimental and the bioinformatics together really nicely. Thank you, Amanda.
good, I'm happy to hear it. Okay, so let's move on to these next questions. Hopefully these questions are a little bit easier. Now they went through a bit more detailed explanation, but we'll see. So again, I know it takes a while to wrap your head around these BAM files. So next we have how many reads align in the same orientation relative to the reference, and what about the opposite orientation relative to the reference, and how do you know? So Lisa, if you could put up that next question, and then I'll give all you guys a minute or so to answer it. So I got 5-5, 2R1 map the same orientation as a reference, 2R2 map same 1-1 sign a template length column, 2 forward, 2 back, have positive negative, 2 reads as they start in the same position, 2 reads as they start in the same starting position, 2 reads same, 2 reads opposite. Yeah, you guys are right overall. So you're correct that there's only 2 reads aligned here. So how you think about it is up to you. So I know there's only 2 reads aligned. Basically, all that you're looking for is in this template length. So this is the template, again, that you're going to have a forward and a reverse read with respect to each template length. And so if the read is in the same orientation relative to the reference, that template length sign is going to be positive. If the read is in the reverse orientation relative to the reference, that template length is going to be negative. So for mate pairs, you're always going to have one in the positive and one in the negative direction. So depending on how you look at this, if you're just saying, oh, we only have 2 reads here, and so we're going to have reads aligning in one mate of the read pair is going to be in the same orientation, and one mate of the read pair is going to be in the reverse orientation. That's a fine way of thinking of it. I think of it as each read, which there are, of course, only 2 mate pairs here. And so 1 mate pair is in the forward, 1 mate pair is in the reverse. Again, the sign of that template length is there. If you want to do each read relative to the transcript, so we know we have 5 transcripts here. And so we see 5 reads aligning in the same orientation as the transcript, right? So this read is aligning in the positive, same orientation as this transcript. It's mate pair aligning in the negative. This read, which is the same read as above, but this time aligning to a different transcript in the same orientation, it's mate pair aligning to the reverse, the opposite orientation of the transcript. And so you can go down like that, and then we have the 3 transcripts that the other mate pair is aligning to. And again, one mate in the positive direction, and then its mate pair is going to be in the opposite direction with respect to the reference. So yeah, you could take either. Your mate pairs with one being in the same direction, one being in the opposite direction for each transcript that it maps to is fine. Or if you wanted to take it as a mate pair to transcript pair, then we have 5 transcripts represented, 2 reads, and so 1 read mapping to 2 transcripts, the forward read, the reverse read mapping in the opposite orientation, and then same with the other read, but to the 3 transcripts. Any questions about how to find out the orientation of the read relative to the transcript? Okay, great. So last question. Again, we went over this, so hopefully it'll be quick, but if not, we can go over it again. So what are the min and max template length shown? Min and max template length, and how do you know? Another few seconds. So if you guys want to put in your best guess if you're unsure, or just say that you're unsure. Go ahead and show the results. 167 and 230, 167 and 203, 334 and 406, 167 and 203, not sure. Okay. Who did 334 and 406 if you're feeling okay about it? I'm curious how you got those numbers.
That was me. Well, because I took the T length. Went to the T length and it said positive and negative. And I was remembering the figure you showed before, the starting position of the forward read and the starting position of the reverse read, and it showed the total length of the sequence, and that's what I added in the total read. But I honestly,
Gotcha.
yeah, that was my logic. But looking at it now, I'm having second thoughts.
Yeah, so I guess first of all, we know that we only have 2 templates, right? We only have 2 mate pairs, and each mate pair is sequencing 1 template, right? So if we only have 2 mate pairs, we can only have 2 templates here. So we only have 2 templates. It's just that those templates are mapping to multiple locations on the genome, or multiple transcripts. So this template length, so it actually does that calculation for you already, Mark. So when it gives you the template length, it is telling you already the start of read 1 to the end of read 2. So this is what it's telling you as the template length. So you don't have to add anything. I get what you're saying is you're probably saying if these were set up a little differently. So let's say that read 1 was here and read 2 was here. And so if you're thinking the template length might be from the end of here to the end of here, and then you add in the size of the reads. I don't know if that's what you were thinking.
Yeah, that's what I was doing. This is a lot to take in. I'm trying to piece it all together.
Yeah, trust me, I know. So if it was something like this, so in the original example I had read 1 and read 2 overlapping. So in this case, this original case, I had basically the template be what I would hope it would be. Like I like to see a little overlap between the reads, but that doesn't always happen. Sometimes you have longer inserts that you're sequencing, in which case you could have your read 1 start over here, and you could have your read 2 be over here with some space in between. If that was the case, that template length would calculate still from the far left side, or the 5' side of your read 1, all the way through the end of your read 2. So if you had something like this, the template length would be from, again, the leftmost position of your forward read to the rightmost position of your reverse read. So it takes into account the size of both of your reads, and also if there was any base pairs in between your reads.
Okay, I see. I got it. Thank you.
Sure. And so because we only have 2 mate pairs, we only have 2 templates that we're working with here. That's why in this T length column, What you're seeing is either 167, and again, positive being the same orientation with respect to the reference. So this read is going to be in the positive orientation with respect to the reference, negative being in the reverse complement orientation with respect to the reference, or this one's mate pair. And so this template that sequenced using this mate pair is 167 base pairs long. And so this, right, if we're sequencing, if each one of these reads, I didn't do, like, you can do a character count on these reads. I don't know if they're the same length or not. But let's say that they are the same length, and if they're both 150, so they're going to be 150 in one direction, 150 in another direction, and the total length is 167. So that example, they're pretty overlapped, right? Because if they were both 150, let's say, and your template is only 167, then that means that there's a lot of overlap between your forward and your reverse read. Because if it's only 167 long, and if these reads are indeed 150 base pairs each, 150 base pairs in this direction, 150 base pairs in this direction, and so that only leaves 167 minus 150. So it's only 17 base pairs in addition to the part that overlaps. So it might be some additional here and some additional here that isn't covered by read 1 or read 2. So your template would be shorter. So we have a lot of overlap here. So this in the other mate pair that we have is depicted in the last 6 lines. Again, this template or this mate pair, which is sequencing this template, the forward and the reverse read for this 167 length template, is mapping to 2 different transcripts. And then we have this other mate pair here, whose template length is 203, mapping to 3 different transcripts on the genome. And so again, 203 is the template length here. So the range, or the min and max, the min would be this first mate pair that's shown up at the top, which is 167 insert length. And then the second mate pair that's shown in these first 10 lines has a length of 203. So this tells me that this insert or template was slightly longer. It was a longer piece that was sequenced than this one. Does that make sense? Any questions about that?
No, that actually cleared it up that cleared it up a lot. Thank you.
Great. Anyone else still having trouble wrapping their head around this?
Are you going to cover the intervening, how to interpret that 149M? Is that the cigar string?
Yeah, so the 149, I mean, we can. So 150 and 149M, that is actually the read length that is an exact match. So M stands for match, and then this is the number of base pairs that are an exact match. So this confused me. I don't know, maybe one of the other instructors will know this. So I would assume that this would have different quality score, like quality map scores here, because, oh no, because this, never mind, because it's the same template. But so for this read, the forward read, so the position that it's sitting on in the reference, it has 150 base pairs, so it must be 150 base pairs long, since that's the max length of a read, that matches exactly to that location on the reference. So all 150 base pairs of this forward read are an exact match to that location on the reference. And for this one, it says that 149 base pairs are an exact match. So that either means that there's one mismatch, but because they have the same quality score, I wouldn't think that, or it means that this read length is 149. So we could see that. Open it up in Word, I guess. I'm just opening this up in a Word document that I have up on another screen. Do a count. There are 11 parameters being shown. Each of the parameters explain how this is pronounced. Yeah, they're actually 12. So this last column, 12 and anything plus are additional like SAM attributes. There's always going to be those first 11 columns. And then depending on if you added SAM attributes to it, you might have a 12th column, which will have that information. And there are some other options you could add, which might give you some additional columns in the BAM file. But they'll always have those first 11.
And then the Phoenix?
Say that again?
Then there's the RNAseq, it says the reference is the same, so it was equal. I guess that refers to the next read.
Yeah, so that's another
And
indication.
so they're equal.
Yeah.
And then what about the P next, position of the mate, 1 minus base position?
So it's one minus the one base position of the next aligned read in the template. And so if we
Puts it on 630.
where are we at? Which one is this? Something on that. So yeah, so 630, and you'll see that so basically it's looking across your reference genome, and wherever this mate this read ends, it's looking for where's the position of the next read that aligns to the reference genome. And so the next read that aligns is going to start at 630. And what you'll notice is if you look at the next
Yes, it's
below it,
complementary.
starts
Yeah,
at 630.
perfect.
Right. And so this one
That
is
helps. Yeah,
the next aligned read that it's finding,
Yeah.
it's mapping to
612, the
612,
previous
and
one.
then you see it. Yeah, the next aligned read
Yeah.
is 612.
So they're matching up. Great. Thank you, Amanda. Thank you.
Sure. And then I did just check, this is 150 base pairs, so every single one of them is matching exactly. Let me do its next one. Yeah, and this is 149, and so all 149 of which is the length of the read, this reverse read is matching. And so this one here, again, it's the same read, so you should be the same length. And since all these are matching, we do show again that all 150 base pairs are matching to this transcript for this read, and its mate, all 149 base pairs of the respective reverse read is matching to this transcript. So that makes sense of why the mapping quality is the same. And you see the same for this next mate pair too. So both reads in this next mate pair are 150 base pairs long. So the forward read is 150 and the reverse read is 150. And I know that it's 150 because they're all, the max that it could be is 150. So if this number is 150, then nothing was trimmed from it. And then M means that all of them were matching. So if this only had like 140 of 150 matched, it would say 140 would be matching. Any other questions about any of the columns or how to interpret these BAM files? Okay. Let's see. Now we're going to take a look at the log final out file. So I did go through this a little bit in the lecture. And so we're going to take a look at the log final out for this first sample that we're analyzing, flight rep 1. And we're going to answer some questions. And hopefully I'll probably cut you off after about a minute so that way we get through all these questions before we get into our break. So go ahead and run that command. And you should see something that looks similar to this. So again, if you remember from the lecture, this is a summary of what happened in the mapping. So overall, of all the reads for this samples file, what did the mapping rates look like? So, and it's nice, it gives you some timestamps up here. It tells you the number of reads that it was working on, the average input read length. So the average input read length, the average template length, and then uniquely mapped reads. It tells you the raw number and the percentage. It tells you information about splice sites, both annotated as well as non-annotated or non-canonical. It tells you stuff about the number of reads that map to multiple locations, as we saw in the top of that BAM file that we looked at. It also tells you if, for the unmapped reads, a little bit of information about why they might have been unmapped. And so I want you to take a look at that file and start answering some questions. So Lisa, if you could put up this first question, which is how many reads map to a unique location on the reference genome? So how many uniquely mapped reads do we have? How many uniquely mapped reads are there? Give me the number or the percent. And that information is all in this log file that we just opened up with the cat command, which is one of the outputs from the star alignment command. How are we doing on answers, Lisa?
We have 7.
Okay, I'm gonna go ahead and show. If those of you guys want to put in a quick guess, if you don't know. Okay, let's go ahead and review. A lot of right answers. Yeah. So the only one that's off is this one. So let's go over real quick where we find this information. So how many reads map to a unique location on the reference? We're looking for uniquely mapped reads. And so right here in the log final file, there's a whole section that talks about the unique reads. And so if you want the number, it's the first line underneath that section, which is the uniquely mapped reads number. So this is the total number of reads that map to a unique location. And if you wanted that in percent, it also calculates the percent of total reads, which is essentially the number of uniquely mapped reads divided by the number of input reads, or the number of reads that were aligned. And so we see that 52,772,596 reads mapped to one location only on the reference genome, which makes up 77% of the total reads, which is great. For the person that didn't get that, is this clear now where that information was found? Okay, I'll take silence to mean yes, perfectly clear. Okay, next question here. How many reads map to multiple locations on the reference genome? So how many multi-mapped reads do we have? So Lisa, if you could open up that question. Okay, guys, in the interest of time, I'm going to go ahead and show those results. So how many reads map to multiple locations on the reference genome? Yeah, everyone who answered answered correctly. For those of you who couldn't get an answer in, if it was because you were unsure, there's this whole section after the unique read section called the multi-map reads. And this gives you information about reads that were mapped to multiple locations. And so the first number listed here is a number of reads that map to multiple loci or multiple locations on the reference genome. So that's where we got that number, which is 8,054,902 reads that map to more than one location on the genome, which is about 11%, a little over 11% of the reads map to multiple locations. Is that not clear to anyone? Okay. We are at 1:30, and I do want to be respectful of the break time, so you guys can all go ahead and take your 20-minute afternoon break, and please be back here by 1:50, and we'll get started with these next 2 questions.